So I'm Amy. I'm a third-year PhD student at the University of Birmingham. I work in the Jackson lab, and all of us are working pretty much on tree pathogens, but this ranges from fungal pathogens to bacterial pathogens. So as you all know, the endemic cloud forest is along the peaks in the centre of St Helena, with many of the endemic trees seen along the central ridge. So here we have the three main peaks, Mount Actaeon, Diana's Peak, and Cuckold's Point. And in my project, I'm focusing on looking at the endemic tree species and the disease that is impacting them. So this is just some of the species that we're looking at. So the she cabbage, dogwood, whitewood, black cabbage, and the he cabbage. So why is this work important? There are many threats to the cloud forest. So the first and foremost being that I'm looking at is an unidentified disease causing trees on the peaks to die, but also in the endemic nursery and the gene banks. This is a major concern as there's already been some extinction events, and many of these tree species are threatened by low numbers. So this is a particular concern as many of these tree species are keystone, especially the black cabbage tree. And these are really important for the island's water capture, their contribution to capturing carbon, the support of other endemic biodiversity such as lichens and invertebrates. And also with this disease comes the other associated risks such as movement of the disease to different species. And some of the reasons that this work's really complicated is because other impacts could be affecting the health of the trees, such as the deterioration of soil after grazing and the plantation of flax across the peaks, and also climate change increasing the susceptibility of the endemic trees to any newly introduced pathogens. So here I've got some data from the weather station on the island, and as you can see there's been a gradual decline in rainfall from 2001 until 2023. This isn't significant, but it has a downward trend, and we see the opposite with temperature, where we see from 2001 to 2023 that temperature has gradually increased. So the main aim of my project is to identify pathogens associated with the endemic trees, to better understand to improve our understanding of the pathosystem, and use this information to try and help inform management decisions and safeguard the cloud forest. So to break that down into some objectives, this is specific to my PhD project. The first initial goal is to identify some candidate pathogens in a survey of the forest microbiome, monitor black cabbage trees in particular over time, explore the pathogen host range through inoculation experiment, which Jane will be talking about a bit later, and then a bit more detail. I'll be doing some molecular work, so we're going to characterize the pathogen by developing some novel reference genomes for them from the isolates from the island. Look at designing a qPCR diagnostics tool and monitoring, and see whether we can monitor how the pathogen's been moving around the island through a population genetics study. So for this talk, I'm just going to focus on the first three objectives. So some of the symptoms we're seeing in the cloud forest, as well as the gene banks and the nurseries, is this unusual tip dieback, where we see some tips which look like they're dying, and we cut into them and we can see this necrosis inside and this black staining. But more importantly, we're seeing this collapse of trees, both mature, semi-mature, and saplings, where they wilt and lose their leaves. We're also seeing this sort of black canker around the base of the trees and into the root system, where we're seeing a lot of trees have completely lost their root system. And then eventually we're seeing the complete death of those trees. So how do we tackle identifying candidate pathogens? We take our tree, and the symptoms of wilt and sudden death is suggestive that this is a root pathogen. Like we say, we've seen these lesions on the root. So we dig down, expose the roots, and as you can see, where there's a black part of necrosis and a bit of the tissue which is still alive, we cut along that leading edge, and we plate that onto different media to try and extract the potential pathogens from the necrotic and healthy tissue. So we're using a specific medium called PARP, tap water agar, which is a low nutrient rich medium. We're also basing things with apple and using soil. So once we've isolated from the tissue, we can identify those isolates further using two different techniques. So morphologically, looking at structures such as the spores, but also through molecular methods. And here I've got an example of one of the common ways we've been testing these isolates. So we will sequence the ITS, which is a conserved region in most fungal, eukaryotic organisms. And we can use this and align it to a database to see what our culture is. So this is an example of a phylogenetic tree that I've produced using several isolates from the island and other species. So we've got here Phytophthora from the clade 8A, our island isolates, and Ilianectria from the radicola complex and our isolates. So you can see nicely in green that the island isolates cluster with Phytophthora calmanii. However, the isolates from Ilianectria, which is another fungal pathogen, do not. The island isolates cluster with different species. So it's possible that these have either been classified incorrectly, or they are so closely related they're hard to separate, and it could be that there's multiple species that are acting in a complex together. So moving on to talk about the main part of my work, which has been monitoring of black cabbage tree health. This is something that's been supported by Zach and people from the peaks. So I've got 12 black cabbage trees which are representative of the central ridge, and we've been monitoring these for 2 years now. So we're looking at the height and the crown cover, the percentage of symptoms we're seeing. So crown loss, the wilting of leaves, leaf drop, and leaf discoloration. We also have some other measurements such as the size of the tree and the abiotic conditions. So this is temperature, humidity, wind. And as you can see, the most interesting thing that we've seen is the death of 2 of those black cabbage trees in the monitoring period. So I've separated the trees based on 50-meter intervals. So we have trees at a low height of 700 meters to 750 meters, trees at a mid height of 750 to 800, and trees at a high from 800 to 850 meters. So we can see that there's been more loss of crown in the trees at a high interval and a mid interval. And you can see these dots which represent dieback events, and these high bars are showing the 2 trees which are now deceased, which is black cabbage tree 7 and black cabbage tree number 9. So if we look at these in a bit more detail, along the bottom we have the time, so this is each month that the data has been collected, and up the side we have the percentage of crown loss. So these are our low down trees. We see there's some fluctuations but nothing significant. When we look in our mid trees, we can see that tree number 10 has had a major loss of crown, followed by recovery, which is now stable. And that blue tree, which is high up, is tree number 4. Since monitoring began, this tree has lost half of its crown, but has remained stable, and it's likely this loss of 50% crown cover was probably due to something non-pathogenic. However, when we look at our high trees, which are where the 2 now deceased trees are, we can see that before succumbing to complete dieback, we have this period of crown loss followed by recovery, but not quite recovering fully, and then followed by another increase in crown loss. So it appears there might be a period of time that the trees are able to recover, but once this threshold has been reached, they can no longer recover. So some of the other work we're doing is, like I said, looking at the discoloration of the crown, the wilting, and the leaf drop. So I just want to focus in on the wilting. So as you can see here, what I've done is scored the severity of each symptom on a scale of 1 to 6, 1 being not very severe, 4 being very severe, and 6 meaning the tree is now dead. So we can see on the wilting scale that trees 7 and 9 have 2 episodes of wilting before the tree dies, and this is what we're seeing commonly across the peaks in all the endemic species. So we have this episode of wilting, and then after that the leaves are lost, and then we have complete dieback of the tree. So that's it from me. I'm going to pass over to Jane now. Just want to thank everyone for being here, and for my collaborators and the St Helena Research Bursary, which has given me the opportunity to be here again and keep monitoring and working on the pathogens. So this is everyone, and thanks for listening.
So this is the second year. We're just coming to the second year of the project now. So I just wanted to give you a quick update on the findings we've had so far. So Amy's work feeds very much into what we're doing as well as a part of it. So she's covered a lot of the background information. The main project is the Darwin Plus. Sorry, Kirsty, it was 157, not 137. Managing the pathogen threats to St Helena's biodiversity. But as part of that, there was also the food security part. So we were looking at the crop plants as well, the farmers are growing, and other things people are growing on their land as well. So there was the 2 aspects, the endemics and the crops. I'll just say a quick thing about the crops. We didn't find very many diseases here. There was the common things like late blight on potatoes, tomatoes, but it was all very well managed. The farmers and the growers knew, some of the issues, and they were taking precautions against those. So that's the good news. And then the other part, we didn't really anticipate there would be that much to do, really, but was the endemics. And we found some issues in the first year, which we've continued throughout the project with. So there was some significant dieback in the trees on the peaks, so the endemics, which are the whitewoods, the dogwoods, the she cabbages, he cabbages, black cabbages, the bastard gumwoods, and the redwoods. So there was some occurrences where these trees had started to die, and we wanted to look into that a little bit further. I should say a little bit about myself as well. I've been with CABI about 22 years now. I'm a plant pathologist, so I've always studied plant diseases. I do a lot in agriculture now, but mainly I worked on tree crops, cocoa, coffee, bananas, not really a tree, but, similar. So this is the reason why I was sort of drafted in to do a bit of work on this, seeing as there was a lot of issues with the trees. So this rapid demise of the trees, significant threat not only to the tree species themselves, but also the other biodiversity that is supported by them. So a lot of the insects that use them as a habitat, or as food. So you can tell this has been written by an entomologist and not a plant pathologist. So this is Norbert Mackenzie, who's the project lead. He's an entomologist and has worked on a few of the projects in St Helena before. So we're looking at doing a sort of eco approach to this and thinking about the environment as well as just the trees on their own. So we started investigating the trees, and the first thing we saw really was the whitewoods. This was very much led by the team up at Scotland and Rebecca as well, pointing us in the right direction. So we were seeing, this was very common, what we were seeing. You can see there's very few leaves left on the trees. What leaves are left on there starting to wilt, and it was quite sudden. So the first thing we did was to sort of have a look at the tree, and as Amy said, this is quite typical of a root pathogen. So we'd go to this region. So what we really want to see is the roots of the tree, but it's very difficult in situ. So the best place we can start looking is in this collar region where the soil meets the tree. And you can see here, it's all black and really not very nice. It doesn't look very healthy. So the next thing we do is take a knife or a machete and we take away some of the bark. And what we were finding was we could basically pull this off with our fingers. It was just completely rotted. And the wood underneath is very hard, which makes it difficult to sample as well. But you get this dark stain in here. So healthy wood should be very white or cream coloured. And we can see this sort of dark red or black staining where the wood should be healthy. So first of all, first sign's the wilting. Then we get this rapid collapse, which happens quite quickly. All the leaves fall off. And then we can see this rotting and discoloration sort of at soil level. So one thing we did have at the time were these LFTs or lateral flow tests, a bit like a COVID test. You can see it on the far photo. And we can take a piece of the tissue and you shake it up in a bottle with some ball bearings in it and you pipette it on the strip. And you get 2 lines if it's positive for Phytophthora. So any go-to root pathogen, Phytophthora, we would consider as an issue. It's common and it causes these type of symptoms. One disease you've probably heard of is late blight and the potato famine. That's a Phytophthora, which has devastated potato growing regions all over the world. So that affects plants both above ground and below, but many Phytophthora just affect below ground. They're soil pathogens, so they affect the roots. So this was quite a nifty little device to give us some idea of what was going on, but we rapidly discovered it didn't just pick up Phytophthora, it picked up some of the other weak pathogens or soil organisms. They're the pythiums, so it wasn't as good as we hoped, but it was a start. Right, this is a very busy diagram. So we carried on isolating and we took the samples home and we molecularly identified them, and it was looking very likely it was what we call a oomycete or a water mould, so Phytophthora falls into that group. So many Phytophthora species, as I said, are known to be plant pathogens, so it was a good bet. And as we sequence more and more isolates, they're falling out as a Phytophthora kelmanii, that's the species name. Unfortunately, not much work has been done on this organism, so there's not much information available. So this circular diagram here represents all of the Phytophthora species. So this is the whole genus Phytophthora. So there's probably about 200 different species in there. So somebody's molecularly ID'd all these and then grouped them together, so the most related ones fall out. And this is where we find Kilmanii, and we call this a clade. So these are all very closely related. Amy mentioned this before. This particular one is called clade 8A. So this is where we're finding our proposed pathogen. So mainly on whitewoods, but we've also found it associated with she cabbage and black cabbage and possibly the dogwoods. So at this stage, we can only say it's an association. We can't say this is definitively the pathogen that's causing the disease. We find it there. It's from a known group of pathogens, but we have no proof. So next steps. I just wanted to mention this. This is the life cycle. So it lives in the soil, it affects the roots, it produces a lot of mycelia and fruiting structures, so it reproduces this way and combines. It can also go from root to root, so where the trees have wide root systems, it can jump onto the next tree. But really I wanted to highlight this because it's in the soil and it spreads through moisture and water. When the soil's muddy, it's transferred a lot by footfall, so on people's shoes where you transfer mud and soil, and in vehicles, tools as well. So any movement of soil or infected water can move the pathogen, hence restricting access to certain places where we know it is. So next step, so we've taken a lot of samples, we've found this pathogen, or what we think is the pathogen, Phytophthora. As a plant pathologist, you then need to prove that this is the organism that's causing the disease. And we do something called Koch's postulates. So Robert Koch was around in the late 18th century when he came up with these postulates. He was a clinician and a bacteriologist. He wasn't a plant pathologist. So these have been developed for human diseases, but they're also applicable to plant diseases as well. So nowadays, we usually call it pathogenicity testing. So we want to know if that pathogen, we test it to see if it causes the disease. So when a plant becomes infected with a microorganism, it can be a bacterium, a virus, fungus, or a pneumycete. We say it's, if it's susceptible to infection, we then look at the postulates. So the first one is the organism must be consistently associated with the diseased part of the plant. So as Amy explained, you take your sample from the leading edge. So that's what we call an isolation. We isolate the pathogen. Then we take it and we grow it up on some culture medium, a little plate with something the Phytophthora likes, so potatoes, carrots, something like that. And then we grow it up and it forms a nice fluffy white culture. And then what we need to do once it's in pure culture is take that isolate and put it back into a healthy plant. So we've had some seedlings, I'll show you on the next slide. We've taken the fungus, we grew it up on some rice, and it colonises each grain of rice, and we put the rice into the soil with the healthy seedling, and then we wait. So what we want to do is reproduce the symptoms we saw in the field, so the wilting, the root rot, and then we isolate the pathogen back out of the seedling, grow it up in pure culture, molecularly identify it again. So if it's the same thing, then we have proof that that has caused, we've put that in, it's caused the same symptoms, we've got it back out and identified it. So this is what we have to do to prove that a potential pathogen is actually causing that disease. So this is just a representation. So this is what we've been doing in the lab. It's been very exciting. So at the start you have your infected plant, then you have the diseased part and the healthy part. We take a little section from there. We surface sterilise it to get rid of any bacteria, fungi on the outside, and then we put it on a little plate with something that we think the pathogen will like, and then give it a few days and it starts to grow. We then take a little square, put it on a new plate, and there we go, we've got a pure culture. So then this goes for identification. So in order to do this, at Scotland, the staff there very kindly built us a new facility. So to keep it away from the nursery, it's a semi-quarantine. So it's isolated from anything else, and we can work in there and keep the pathogens separate. So this was finished off in January this year. So at the moment we have whitewood seedlings and dogwood seedlings. So there's a picture of them here, growing in pots, completely separated, so any water going in is caught in a dish, so we don't get cross-contamination. So the team that came out in January set these up with the staff at Scotland, inoculated them, then we've waited about 5 weeks, then Amy and I have come back to see if there's been any disease symptoms. And this is what we found. So 5 weeks after inoculation, this is the control plant, so no Phytophthora in there. This is it when we've taken the soil from the roots and washed them. It looks nice and healthy, no wilting, nice root system. 2 of the treated plants. So we did it in 2 concentrations. The first ones we put 5 grains of rice in, colonised with the pathogen. And then the second one, we put a much higher inoculum in, because we don't know how much this is going to take to affect the trees. So these ones had 15 in. And you can see when we went in on Sunday just to have a quick peek, they'd totally collapsed, totally wilted. So on Monday when we came in, we took them out. You can see on this one, with the lower level of inoculum, the roots have gone black, they've rotted, there was very little healthy root tissue there. And this one is in a bad way, there's no roots at all. So they've struggled on with very few roots, but they look healthy up until a point where they just collapse very quickly. So what we do then is we take them into the lab, and then we basically cut some sections from the roots, and as we did in Cox postulate, plate them onto some media. And that's what Amy and I have been doing today, looking to see what we've got. But fungi look very similar, and oomycetes when they're in culture. So we've purchased this whizzy piece of kit, which is relatively new. It's a bit of a mouthful, and we call it LAMP for short. And somebody very wittily at Optogene has called it the lamp genie. So this is used in the field sometimes, mainly in the lab. It can be a basic lab. And it's loop-mediated isothermal amplification, so a molecular technique for basically amplifying samples you put in, and then you test it with specific markers. So there's been lots of work going on in the UK to find a piece of the genome or a piece of the gene in this particular pathogen that's unique to that pathogen. So in the reaction we can add it and it will amplify it, and then it will give us a nice line if it's there. So this has taken quite some time. We're not quite there yet. We have it specific to this group here, which is pretty good going. These aren't particularly common pathogens, so we're getting close. But the benefits of this are, it's relatively cheap, the reagents are relatively cheap, and it's very quick. It takes 30 minutes, 10 minutes to set it up, and then 30 minutes to run it, and we get a positive or a negative. So we're using lots of standard Phytophthora species as well to compare it against. And we can use it on pure culture, so those little plates we have with the isolations on, and we've just been looking at some of the diseased roots. So we've been actually just taking some of the roots and putting it into what we call a DNA extraction kit. You can buy and order from online. So it takes about an hour to go through that process. It's a good quality, but a little expensive. So further investigation is needed on soil and woody tissue. So the samples we take up on the peaks and at George Benjamin, it's very hard woody tissue. The roots are relatively soft and easy to extract from. So we need to look at other methods for those. The soil will be brilliant if we can manage to identify the pathogen in that. Then we can look over different parts of St Helena to see if the pathogen's in the soil. But at the moment, the kits aren't particularly helpful and we're looking for other methods. So going forward, we've another year left on this project. We've also had a bit of additional funding from DEFRA, which has been really good, and the Cloud Forest project as well. So going forward into the final year, we think it's imperative that we continue with the Cox postulates or the inoculation experiments. So we want to reproduce these symptoms in other tree species. So we can only do that as and when they're available. So we've done whitewoods, we've also done dogwoods, and we didn't find any symptoms in the dogwoods yet, so they're still up in the shade house at Scotland. The other thing we need to look into as well is the current spread. So again, developing methods to find the pathogen in the soil so we can test different areas. Monitoring, regularly monitoring the decline or the effect on the plants. So every month go out, take notes on which are showing symptoms, just so we know how rapidly this is spreading, if it's spreading, or if it's quite a slow process. And then the final point was the development of long-term management plans. So looking at other countries, other groups where Phytophthora has been an issue, like what can we do? Better practices in the field, because we can't manage this with chemicals. It's a soil pathogen, it's very difficult. It's one of those things we just need to manage. And if it's only in certain areas, looking at restricting movement from those areas into new areas where the pathogen presently isn't. So that's a really important one. So that's all I have for you at the moment. Thank you very much.
I was just wondering, did you find any other potential candidates to...
Oh yes, I forgot to mention that. Yeah, we found a pathogen called Ilianectria, so this is a tree fungus. Amy touched on it before. It seems to be in a complex where there's lots of different species, or there's not a lot of work being done on it. And so it may just not be very well described. So in the shade house, we've also inoculated the whitewoods with the Ilianectria, and we've had no symptoms yet. So whether that's something that moves in as an opportunist when the Phytophthora comes in, we don't know. So yeah, we're looking at other groups like Fusarium as well. They're well-known plant pathogens. The problem is when you isolate from trees, there's things called endophytes in there, which are fungi or bacteria that live in the tree. So we isolate those out. They don't cause any diseases. And there are all these other pathogens and secondary decomposers, really. So we end up with this soup of different fungi, different organisms, and it's really hard to pull out, what's causing the disease. This is why it's taken us quite some time, really. So you have your candidates that you think it can be, but you've got to remain a bit objective and see how things pan out, lots of sampling. But it's incredibly difficult to work with Phytophthora. I was just saying to Rebecca today, I don't know how it causes disease in the wild because it's so hard to work with in the lab.